My name is Maddison Arsenault, and currently I'm studying pig fecal microbiota during early stage of production. So within the gastrointestinal tract of swine, there are thousands of microorganisms that form a complex microbial community. These microbes interact with each other and the host pig to provide essential metabolic and immunological functions. Targeting the microbiota in early life by promoting the development of beneficial bacteria is a potentially powerful tool to improve swine health, increase growth, and decrease production costs. The objective of this study was to understand piglet fecal microbiota by defining its composition and its progression from birth till 1 week post-weaning. This was a longitudinal study of 9 cohorts, each containing 40 pigs. Each cohort was located at a distinct farrowing source across Ontario and Quebec. 4 fecal samples were collected from each of the 360 pigs in this study, and the sampling ages are summarized below in the bottom left table. In order to identify which bacteria were present in the microbiome, the V3, V4 regions of 16S rRNA were extracted from the fecal samples and then sequenced with MiSeq Illumina sequencing. Following this, sequencing data from 978 of the 1,440 samples were run through a bioinformatics pipeline to provide preliminary results. So, looking at figure 1, each point on this graph represents a fecal sample, and the distance between the points represent microbiota similarity, meaning the closer 2 points are on the graph, the more similar their microbiota composition is. Overall, 3 stages of microbiome succession were identified, and each stage was associated with a distinct community cluster. So we can see these 3 clusters grouping on this biplot, with cluster 1 on the left in black, cluster 2 in the middle in yellow, and cluster 3 on the right in red. Figure 2 then examines the age of the pig at sampling along the x-axis. And note this graph is faceted, so pre-weaning samples are on the left, and the distribution of post-weaning samples is on the right. This figure highlights the prevalence of each cluster at different sampling ages. Briefly, cluster 1 in black occurs mostly during the first week of life, and then all other pre-weaning samples were classified into the second yellow cluster. Interestingly, cluster 3 almost exclusively appears in post-weaning samples, and this highlights the shift that occurs in microbiome community composition at weaning, regardless of weaning age. Lastly, figure 3 shows the relative abundance of phyla within each cluster. Notably, all 3 clusters are dominated by Bacteroides, Firmicutes, and Proteobacteria phyla. Cluster 1 also contains a high relative abundance of the Fusobacteria phylum, but in contrast, cluster 2 and 3 contain low levels of many different phyla. So, microbiota community diversity increases as the pigs age and progress from one cluster to the next. Overall, this research highlights that pig fecal microbiota progresses through 3 distinct community types from birth until after weaning. Association should be explored between early stage microbiota and external factors like antibiotic use, rearing conditions, or genotype. Then this knowledge can be applied in meaningful ways across the industry by manipulating gut microbiota in early life to improve health and growth from farrowing all the way to finish. Lastly, thanks to Swine Innovation Porc and other industrial funding partners. And for more information, feel free to review this poster or to reach out to me via email. Thank you.